Welcome back to the Deep Dive. Today we are plumbing the depths of something truly fascinating. It's this complex bidirectional communication system that links your gut right to your mind. We're talking about the microbiota gut-brain axis.
That's right, and it's a field that's just exploded.
Absolutely. For too long, the brain was treated almost like a castle,
Yeah.
isolated, self-governing. But the last decade, maybe a bit more of intensive research has really confirmed this revolutionary truth. The state of your gut ecology, it profoundly influences your emotional regulation, your cognition, even your overall mental health. And we aren't just relying on isolated studies here. We're synthesizing a massive body of evidence. We're pulling from really rigorous, systematic reviews covering roughly 2012 to 2023. And focusing specifically on how alterations, changes in gut microbiota, correlate with psychiatric and neurological disorders.
It really represents a fundamental paradigm shift, doesn't it? We now have to view psychiatric health, stress response, neuroplasticity, not as these isolated cerebral events, but as systemic processes.
It's systemic, right?
Yeah.
Involving the whole body.
Exactly. And the foundation of this whole system is the gut microbiota. It's this vast multi-kingdom ecosystem. We're talking trillions of bacteria, viruses, fungi, archaea, all residing within your gastrointestinal tract.
Trillions. It's hard to even conceptualize.
It is. And while all the members are relevant, the scientific spotlight, well, it remains pretty firmly focused on the bacterial kingdom, particularly the dominant phyla like firmicutes and bacteroidetes. They seem to be the primary drivers of the chemical communication we're really digging into today.
Okay, so if we think about this microbial community like a huge, bustling city inside us,
Yeah.
how does it actually relay its status, its complex operating status, to the command center? Up in our skull. How is that message actually getting transmitted?
Well, the transmission system is the gut-brain axis, the GBA, and it's defined as this two-way biochemical and neurological signaling pathway. It's actually highly redundant, very robust, almost ensuring the messages always get through one way or another.
Two ways, so signals go up and down?
Precisely. And this axis involves four primary interconnected components. Obviously, you've got the central nervous system, the CNS. Then there's this massive specialized nervous network embedded directly in the gut wall. That's the enteric nervous system, or ENS. Also, the body's master stress regulator, the hypothalamic pituitary adrenal axis, the HPA axis. And finally, an intricate network of neuroendocrine and neuroimmune systems. It's all woven together.
Wow. That sounds, well, less like a simple biological connection and more like an entire parallel operating system, almost designed for chemical surveillance and constant feedback.
That's precisely why we often reach for metaphors. It helps grasp the scale. The gut microbiota community itself is increasingly called a virtual organ.
A virtual organ?
Yeah, because of its sheer metabolic output and its systemic influence. And the entire gut itself, frequently dubbed the second brain.
The second brain. I've heard that.
Its influence is just staggering. I mean, it regulates how we harvest energy from food, it modulates the immune system's set point, it controls the release of key hormones, and crucially for this discussion, it dictates the permeability, how leaky or tight both the intestinal wall and the formidable blood-brain barrier are.
Okay, let's really unpack this then. If we accept that this second brain is so critical to our systemic health, the central question these systematic reviews are asking becomes, is there a measurable, a verifiable association between common psychiatric disorders, things like depression, anxiety, autism, schizophrenia, and definable alterations, specific changes in the gut microbiota? And if that link exists, which it seems to, we absolutely need to know the precise hardwired routes of communication. How does it actually work?
And that's exactly where our deep dive starts. Analyzing the actual infrastructure of this communication. We can identify several primary routes, but maybe the most direct and in many ways the most compelling is the neurological pathway involving the vagus nerve.
Ah, the wandering nerve. Right, that big cranial nerve running straight from the brainstem down through the neck and chest and connecting directly to the abdominal organs. It's a very literal cable connecting the two brains, isn't it?
Exactly. And the research here, particularly using these highly controlled animal models like germ-free mice, GF mice, provides some of the most direct evidence of its role.
Germ-free. So mice with no gut bacteria at all.
Correct. Raised in a completely sterile environment. And studies have shown quite clearly that these GF mice exhibit significantly lower basal vagal nerve activity compared to mice with a normal colonized gut.
Lower activity in that nerve cable.
Right. But here's the clincher, the intervention part.
Yeah.
When researchers restored the gut bacteria in these GF mice, basically gave them a normal microbiome, the previously depressed vagal nerve activity completely normalized.
Wow. So the bacteria themselves brought the nerve back online, essentially.
It provides direct, almost causal evidence that the bacteria themselves are essential for maintaining the tone, the function of this major neural highway.
That really suggests the bacteria aren't just sending passive signals up the wire. They're actively maintaining the volume, maybe the bandwidth, of the signal being transmitted up to the brain.
That is absolutely spot on. And the structure, the actual anatomy of the vagus nerve itself, offers this profound insight into the direction of influence.
How so?
Well, the nerve contains afferent nerve fibers. Those communicate from the gut back to the brain, and efferent fibers, which transmit messages the other way, brain to gut. Anatomical studies consistently show that the afferent fibers, the ones going gut to brain, overwhelmingly outnumber the efferent fibers
Outnumber them by how much?
by an approximate 9-to-1 ratio.
9 to 1. That ratio,
Okay.
that implies a pretty radical shift in perspective, doesn't it? It means the brain is maybe fundamentally an organ of response, configured to constantly process this massive amount of surveillance data streaming in from the periphery, rather than primarily just issuing commands down to the gut.
Precisely. The gut is constantly reporting its status, what's going on down there up to the CNS.
That anatomical dominance suggests the central nervous system dedicates far more processing power, more bandwidth, to understanding the state of the periphery. The level of inflammation, nutrient availability, the presence of specific metabolites, more power to understanding that than it sends back in terms of direct control commands.
Yes. The gut-brain axis, you could argue, is inherently bottom-up in its sensory input based on that structure.
Okay, so moving from that main neurological cable, the vagus, let's look at the local network, the enteric nervous system, the ENS.
Yeah.
The so-called brain within the gut.
The ENS is massive and truly autonomous in many ways. It's a structural network made up of anywhere from 100 to 500 million neurons. That's more neurons than in the entire spinal cord.
Wow.
And it stretches the entire length of the GI tract, esophagus to anus. It operates with its own sensory neurons, motor neurons. It's capable of regulating gut motility, fluid secretion, localized blood flow, often entirely independently of the CNS.
A brain unto itself, almost.
Pretty much. But what's truly fascinating for our discussion today is the discovery that the development, the renewal, and the fundamental function of these ENS neurons are heavily controlled by the gut microbiota.
Wait, so the bacteria aren't just influencing an existing nervous system, they're actually involved in building and repairing the nervous system of the gut itself.
Absolutely. The evidence for this is, again, quite robust in those germ-free models. Studies showed that the neural stem cells residing in the gut of GF mice were demonstrably unhealthy. You could see it by a deficiency in crucial cell proliferation and renewal indicators, things like nestin and Ki67.
Okay.
But when the gut was recolonized with a normal microbiota, these stem cell functions were completely restored.
So the microbes kickstart that renewal process.
It confirms the microbiota is essential for maintaining a healthy, dynamic, plastic ENS. Furthermore, we know a lack of certain microbial products, particularly those short-chain fatty acids we'll talk more about, can lead to a measurable, observable loss of enteric neurons. It really highlights the direct dependence of gut nerve health on microbial metabolism.
Okay, that's the direct nerve connections. Now let's link this infrastructure back to our deepest emotional life and the body's primary stress response system, the endocrine or stress pathway involving the HPA axis.
Right, the hypothalamic pituitary adrenal axis. This is the master controller of the stress response. It determines how we perceive and react to threat, whether that's physical or psychological. And the gut microbiota is intrinsically linked to both the development and the ongoing function of the system.
And we know stress itself impacts the HPA axis.
Oh, definitely. We know chronic or acute stress activates the HPA axis. That leads to prolonged elevation of glucocorticoid secretion. You get measurable higher cortisol levels. And that's a hallmark, a common feature found in major depressive disorder, MDD.
Okay, but what's the key connection here? How does the gut step into this HPA stress cycle? Where's the feedback loop?
Well, stress itself is almost immediately destructive to the gut barrier. That's a critical point. Stress alters intestinal epithelial permeability. It loosens the tight junctions that normally keep the gut lining sealed. This results in what's widely, though maybe sometimes inaccurately, termed leaky gut syndrome.
Leaky gut. Yeah.
This defect creates a critical vulnerability. It allows bacterial components and inflammatory molecules, particularly something called lipopolysaccharide, LPS, from gram-negative bacteria to pass through that damaged barrier and enter the systemic circulation.
Right, LPS is like a red flag for the immune system, isn't it, even at low levels?
Precisely. Once LPS is in the blood, the body treats it like an invasive infection, even if it's low-grade chronic leakage. This influx of LPS and other inflammatory molecules activates a chronic, low-grade immune response throughout the body.
And systemic inflammation. That's bad news for the brain, too.
It is, and it's a known cause of HPA axis hyperactivation. The inflammation essentially rewires the HPA axis, locking it into a state of chronic alarm, which leads to those sustained high cortisol levels. So you end up with this devastating loop.
Let me see if I follow. Stress damages the gut barrier. The damaged barrier leaks inflammatory stuff like LPS into the blood.
Right.
That systemic inflammation then overactivates the HPA stress system, leading to more stress hormones.
Exactly.
Which probably damages the gut barrier further. It's a vicious cycle.
It is. And the microbiota composition itself also changes due to stress. And in turn, the resulting altered microbiota composition then modulates the host's overall stress levels. It feeds back on itself.
This really starts to highlight the chemical communication, doesn't it? The metabolic pathway. This feels like perhaps the most convincing evidence of systemic microbial control. The bacteria aren't just passive tenants sitting near the neural wires. They are tiny chemical factories, churning out these powerful chemical modulators that are biologically active far outside the gut itself.
That's the essential insight, really, of the humoral or metabolic pathway. It's all about the chemicals. We can sort of distill these powerful chemical signals into three major classes that define this microbe-host communication language. You've got short-chain fatty acids, or SCFAs. You have neurotransmitters that are produced or heavily influenced in the gut, and neurotrophins.
Okay, let's start with those workhorse chemicals, the short-chain fatty acids, SCFAs. I know those are acetate, propionate, and butyrate.
That's them. And SCFAs are essentially the end product of fermentation, specifically the fermentation of non-digestible carbohydrates, think fiber, by specific beneficial bacteria.
So you need the right bacteria and the right fuel fiber to make them.
Precisely. And these beneficial producers include some key health-associated genera, names like Bifidobacterium, Faecalibacterium, Roseburia, Lachnospiraceae, Blautia, and Coprococcus. Their roles are just far more extensive than we previously realized. They're not just gut-specific.
Okay, so what are their roles outside the GI tract? How do they influence the brain?
Well, locally, they're essential, of course. Butyrate, for example, is the primary energy source for the cells lining your colon, the colonocytes. It helps maintain that crucial intestinal barrier integrity we just talked about. But systemically, their roles are immense. SCFAs are small, they're lipid-soluble molecules. This allows them to cross the blood-brain barrier, the BBB.
They get directly into the brain.
They do. And once they're in the central nervous system, they exert these profound regulatory roles. For instance, they regulate the maturity and the function of microglia. Microglia are the brain's resident immune cells.
Right, the brain's cleanup crew and defense force.
Kind of.
Yeah.
And if microglia aren't properly regulated, if they become overactive or constantly inflamed, they contribute significantly to neuroinflammation and neurodegeneration. SCFAs help keep them in check.
And is the signaling specific or just a general effect?
Oh, it's incredibly precise. SCFAs don't just sort of wander around randomly. They stimulate specific receptors, G protein-coupled receptors known as GPR41 and GPR43. These receptors are located on enteroendocrine cells, which are hormone-producing cells in the gut lining, and also directly on neurons within the ENS.
So like specific docking stations for these SCFA signals.
Exactly. Think of them like that. They're designed to receive and relay information about the metabolic state of the gut directly up the chain towards the CNS. The presence or absence of SCFAs tells the brain whether the body is, say, well-nourished, whether the immune system needs to be ramped up or calmed down, whether local inflammatory processes are under control. It's critical information.
Okay, so it follows then logically that if the beneficial SCFA-producing bacteria are reduced, which is a state we call dysbiosis, this vital signaling system malfunctions, the brain essentially receives a constant stream of negative metabolic signaling, or maybe just misses the positive signals it needs.
Exactly right. And this is one of the most consistent clinical observations across the board. A reduction of these SCFA-producing taxa and the subsequent low SCFA concentrations that result are general features of dysbiosis observed across a really wide range of psychiatric disorders. It indicates this profound, widespread loss of crucial metabolic signaling that's necessary for both gut health and for neuroprotection.
Okay, that's SCFAs. Now, moving on to the actual neurotransmitters themselves. We primarily think of these chemicals like serotonin or GABA as being made and used in the brain. But you're saying the gut, surprisingly, is the main production site for some.
It certainly is. Let's take serotonin, 5-HT. A staggering 90%, 90, of the body's entire serotonin supply is produced in the gut.
90%, not in the brain.
90%. It's produced by specialized cells in the gut lining called anaerochromophin cells, or EC cells. And importantly, microbial metabolites, including those crucial SCFAs we just discussed, play a critical role in influencing the synthesis of this 5-HT. They regulate the rate-limiting enzyme, tryptophan hydroxylase, inside those EC cells.
Ah, so the bacteria aren't necessarily making the serotonin directly, but their presence and their chemical byproducts, their metabolites,
Yeah.
essentially dictate how much serotonin the gut produces.
Precisely.
So when the microbial environment is unbalanced dysbiosis, again, this serotonin production line gets disrupted.
Exactly. Dysbiosis is directly linked to altered 5-HT production, and that's obviously highly relevant to mood disorders like depression and anxiety. But to illustrate this point beyond just mood, consistently elevated plasma serotonin levels, serotonin in the blood, have been observed in children with autism spectrum disorder, ASD. This suggests that the microbial influence on this serotonin production system extends into critical neurodevelopmental processes as well.
Fascinating. And what about the primary inhibitory neurotransmitter, GABA, the one that calms things down?
Gamma-aminobutyric acid, GABA, yes. This one is produced directly by several common bacterial genera, most notably Lactobacillus and Bifidobacterium.
So they actually make GABA.
They do. And the experimental evidence here is particularly exciting because it shows a functional translation, a real effect. Animal models have demonstrated that administering certain strains, specifically Lactobacillus rhamnosus, JB1 was one studied, can actually modulate GABA receptor expression in key regions of the brain. Modulate means altering the sensitivity or the concentration of those receptors.
So the bacteria, with their products, are changing how the brain listens for GABA signals.
Correct. And this central modulation directly correlates with reduced anxiety and depressive behavior in these animal models. It provides a really clear pathway by which gut bacteria products can directly influence central inhibitory signaling and effectively calm the nervous system.
Okay. SCFAs, neurotransmitters, what a third chemical class? Neurotrophins.
Neurotrophins, yes. Specifically brain-derived neurotrophic factor, or BDNF.
Right. BDNF is crucial for brain health. We know low BDNF levels are essentially synonymous with poor neuroplasticity, poor learning and memory, and generally poor mental health outcomes.
Exactly. And the gut microbiome significantly influences the expression of BDNF in vital brain regions, including the cortex and the hippocampus areas critical for higher thought and memory. BDNF is often called the miracle grow for the brain. It's critical for neuroplasticity, the brain's ability to change and adapt for memory formation, general cognitive function. And as you said, low BDNF levels are widely associated with depression, anxiety, and even schizophrenia.
And you mentioned earlier that SCFAs are involved in this too. How does a simple fatty acid produced by gut bacteria end up influencing gene expression for something as complex as neuroplasticity in the brain?
This is where it gets really interesting. It's an example of epigenetic control. SCFAs, particularly butyrate, act as histone deacetylase inhibitors, or HDAC inhibitors.
HDAC inhibitors. Okay, what does that mean?
So, histone deacetylases, HDACs, are enzymes that normally keep DNA wrapped up really tightly around proteins called histones. This compact structure makes certain genes inaccessible. It effectively turns them off. By inhibiting these HDAC enzymes, hence HDAC inhibitors, the SCFAs essentially loosen that DNA structure. They act like molecular wrenches, opening up the DNA.
Allowing the cell to read the genes that were previously hidden.
Precisely. It allows for the transcription of specific genes, including the BDNF gene itself. This increased gene expression stimulates brain development, enhances neuroplasticity, and offers powerful neuroprotection. So the bottom line is, maintaining that balance of SCFA production isn't just about gut health. It is literally crucial for maintaining the brain's fundamental ability to adapt, learn, heal, and survive.
Wow. That's a direct molecular link from gut microbial activity to brain gene expression.
It is. It's a powerful mechanism.
Okay, so with this mechanistic foundation now established, we've got the vagus highway, the ENS local network, the HPA stress loop, and this complex chemical signaling via SCFAs, neurotransmitters, and influencing BDNF. The systematic reviews you mentioned must provide pretty compelling evidence linking these biological breakdowns to specific clinical conditions.
Overwhelming evidence, really, linking these mechanisms to specific clinical manifestations right across the psychiatric spectrum.
All right, let's walk through some of those. Let's start with the most common mental health challenges: depression and anxiety. The studies found a consistent pattern of dysbiosis. What specific microbial changes were typically observed, and how do they map onto the mechanisms we just discussed?
Well, the finding of significantly altered microbial diversity, meaning fewer different types of bacteria, often a less stable community in depressed patients compared to healthy controls, is, well, it's a rock-solid fixture in the literature now. It shows up again and again.
Lower diversity.
Yes. And crucially, this often involves a decrease in the absolute numbers, the abundance of those key SCFA-producing genera we talked about, Faecalibacterium, Roseburia, etc. This leads directly to the functional deficiency we described, lower SCFA levels.
And that connects back to?
It connects directly back to the HPA axis pathology. Reduced SCFAs mean reduced gut barrier integrity. That means increased LPS leakage into the bloodstream. That drives heightened systemic inflammation, which then feeds that chronic HPA hyperactivation commonly seen in major depressive disorder.
So the dysbiosis isn't just a symptom or a correlation. It seems to be an active contributor to the inflammatory cycle that helps drive or sustain depression.
Precisely. And the reviews also highlighted interventional data that supports this causality. For instance, studies showed that individuals who underwent interventions designed to improve their gut microbial diversity, maybe through diet changes or probiotics, also demonstrated a measurable lower score on the CESD scale. That's a key clinical measure indicating better mental status, less depression.
So improving the gut diversity improved the depression scores.
Yeah. It correlates strongly. It confirms that low diversity is both a characteristic of depression and potentially a modifiable therapeutic target. We also see this mirrored in stress models. Nutritional interventions using specific lactobacillus and bifidobacterium strains show a direct beneficial effect on stress reduction scores in clinical settings, suggesting they can help buffer the HPA axis response.
Okay, let's move to autism spectrum disorder, ASD. The source material confirms that dysbiosis is a persistent comorbidity, often tangled up with those severe gastrointestinal problems frequently reported in individuals with ASD. What does the microbial picture look like there?
In ASD subjects, the typical pattern observed is a clear shift towards a higher abundance of potentially pathogenic bacteria, or at least opportunistic bacteria, and often a significant reduction in known beneficial taxa. But even more specifically, a strong correlation was observed in some studies between a high abundance of a particular bacterium called Desulfovibrio and the severity of autistic manifestations.
Sulfur-vibrio, what does that one do?
They are sulfate-reducing bacteria. Their dominance can disrupt the local microbial community structure, partly by competing with those beneficial SCFA producers for resources. They change the chemical environment.
And this feeds directly back into the chemical language, the metabolic pathways we discussed earlier.
Absolutely. The gut dysbiosis seen in ASD patients is linked to reduced SCFAs, which we know influences microglia function and potentially BDNF. It's also linked to that altered serotonin metabolism. Remember, the high plasma serotonin observed in some children with ASD. That likely traces back, at least in part, to gut dysbiosis affecting the EC cells.
And the persistent GI issues themselves, the constipation, diarrhea, pain, are thought to constantly exacerbate the systemic inflammatory state, which could have profound implications, especially during critical periods of neurodevelopment.
Right, makes sense. Okay, let's talk about eating disorders. These are really complex psychiatric conditions defined by extreme behaviors around food, but also involving significant metabolic disturbance. How does the gut fit into anorexia nervosa, AN, and binge eating disorder, BED? Well, the microbial profiles in eating disorders seem to reflect both the body's altered nutritional state and its inflammatory status. They tell a story. In anorexia nervosa, for example, researchers consistently observe an altered microbial composition marked by a significant reduction in carbohydrate fermenting genera.
The fiber processors again?
Exactly, the ones that make SCFAs. And predictably, this leads to severely lower butyrate concentrations. The gut is literally struggling to extract even residual energy efficiently from fiber. This is often accompanied by an increased proportion of pro-inflammatory taxa, like bacteria from the Enterobacterius family. This confirms that intestinal and systemic inflammation are significant measurable factors in the pathology of AN.
And in binge eating disorder, particularly when it occurs in patients who are also obese. Is it a different picture?
It's a different signature, yes, but similarly pathological in its implications. Subjects with BED, especially those with obesity, sometimes displayed increased levels of certain bacteria like Anaerostipes and Roseburia, which can be SCFA producers. Interestingly, perhaps reflecting higher food intake, but alongside that, a significant detrimental decrease in Akkermansia mucinifera.
Akkermansia. I know that one's important for the gut lining.
Critically important. Akkermansia plays a crucial role in gut barrier integrity. It literally lives in, consumes, and helps maintain the mucus layer lining the colon. So loss of Akkermansia can lead to thinning of that protective mucus layer and increased intestinal permeability, more leaky gut. This dramatically raises the risk of LPS leakage and subsequent systemic inflammation. This loss of barrier function, often linked to reduced akkermansia, seems to be a common thread linking many psychiatric and neurological conditions. We even see a similar reduction noted in conditions like PTSD.
Wow. Okay. Finally, in this section on clinical links, we need to cover the evidence for severe mental illness. Specifically, things like psychotic episodes, first episode psychosis, FEP, and schizophrenia. Is there a gut connection there too?
Yes, dysbiosis is increasingly being identified as a critical comorbidity even in these severe conditions. It's not just about mood or anxiety. Studies show that FEP patients exhibit microbial composition differences compared to controls, and these differences are directly linked to the severity of their psychotic symptoms and also their scores on global functioning assessments.
And what kind of microbial changes are seen? Is it the same pattern?
Disturbingly, yes, we again see a familiar pattern. A specific decrease in key protective butyrate-producing taxa, including Faecalibacterium and Blauschia. It strongly suggests that the generalized loss of the anti-inflammatory and neuroprotective functions provided by SCFAs might diminish the brain's overall resilience, making it more vulnerable to severe psychological distress or breakdown, like psychosis.
That provides a really robust cross-spectrum connection, doesn't it? Linking gut dysbiosis, especially low SCFAs and increased inflammation, across a massive range of psychiatric conditions. Now let's transition into neurodegenerative diseases. You mentioned the role of the gut is becoming exceptionally clear here, potentially moving us beyond mere correlation towards suggested causation.
Yes, this is arguably the most exciting and maybe concerning cutting edge of the field right now. Even if we just look at general aging and cognitive decline, we see GBA patterns potentially determining resilience. The presence of pro-inflammatory taxa like Fusobacterium seems to correlate with decreased brain function over the aging period. Conversely, the continued presence of those beneficial SCFA-producing taxa correlates with improved cognitive function as people age. This really strengthens the overall theory that the inflammatory status, largely mediated by the gut, heavily influences brain status and cognitive decline throughout life.
But the most stunning recent development, it seems, is in Parkinson's disease, PD. The literature appears to be building a really powerful case supporting Brake's theory from years ago. The idea that PD pathology might actually originate in the gut before the toxic protein spread up the vagus nerve and into the brain.
The mechanistic evidence supporting that gut origin theory is becoming highly compelling, especially because researchers have now identified a very specific mechanism involving a specific bacterium that seems to act as a microbial driver of PD pathology. It connects the oral cavity, the gut, and the brain in one quite devastating pathway. They're calling it the oral gut-brain link.
Okay, an oral gut-brain link. What specific bacterium are we talking about here? Which one is driving this?
The identified culprit, remarkably, is Streptococcus mutans.
Wait, Streptococcus mutans. Isn't that the common oral bacterium best known for causing dental cavities? Tooth decay?
That's the one. Researchers found a significantly increased abundance of S mutans, not just in the mouth, but in the gut microbiome of Parkinson's patients compared to controls. It suggests an oral origin, perhaps swallowed repeatedly, that then manages to colonize the GI tract in susceptible individuals.
That's a staggering connection, a potential link between essentially dental hygiene and risk for a severe progressive neurological disorder like Parkinson's. How on earth does a cavity-causing bacterium start affecting dopamine neurons in the brain? What's the mechanism?
Well, S mutans possesses a specific enzyme. It's called urocanate reductase, or ERDA for short. This enzyme, ERDA, converts substances derived from amino acid metabolism, specifically histidine metabolism, into a highly toxic metabolite known as imidazole propionate, MP.
Imidazole propionate, MB, toxic byproduct.
Highly toxic, it seems, particularly to neurons. And importantly, elevated levels of both the enzyme ERDI and the toxic product MP were detected not only in the gut samples, but also in the systemic circulation, the blood of PD patients.
Okay, so the bacteria living in the gut are creating this toxic byproduct, MP, and it's small enough or stable enough to get out of the gut and circulate systemically through the body.
Exactly. MP is capable of entering the systemic circulation, and crucially, it appears capable of reaching the brain, likely crossing the blood-brain barrier. Researchers tested this causality directly in mouse models.
What happened when they gave mice MP?
When they induced elevated systemic and brain MP levels in these mice, the animals developed the hallmark features of Parkinson's disease. This included a measurable loss of dopaminergic neurons, the specific neurons that die in Parkinson's in the substantia nigra region of the brain. They also saw heightened neuroinflammation, the mice developed impaired motor function, and critically, there was increased aggregation of alpha-synuclein protein.
Alpha-synuclein aggregation.
Yeah.
The Lewy bodies. That's the physical manifestation of the disease, isn't it? The toxic clumps of misfolded protein that are thought to kill the neurons.
It is.
So this means a metabolite, MP, produced in the gut by a common oral bacterium, S. mutans, is acting as a direct pathological mediator. It's triggering the core pathology of Parkinson's disease in these models.
It appears to be, yes. It is a phenomenal and frankly quite alarming discovery. But because the mechanism is potentially so specific, bacterium produces enzyme makes toxin causes damage, it allows for highly targeted therapeutic investigation. The studies went even further, identifying how MP exerts its toxic effects at the cellular level.
Okay, so they figured out the downstream pathway. Did the studies identify a way to potentially interrupt this toxic cascade?
They did. Further experiments demonstrated that the neurodegeneration induced by S mutants and its toxic metabolite MP is critically dependent on the activation of a complex cellular signaling protein. It's known as mTORC1.
mTORC1. I know that's involved in cell growth and metabolism.
It is the mammalian target of rapamycin complex 1. It's a fundamental master switch for cell growth, metabolism, survival, autophagy. Lots of core processes. In this specific context, it seems MP acts as some kind of danger or stress signal that excessively activates mTORC1 in neurons. And it's this excessive mTORC1 activation that appears to initiate the destructive cascade, leading to neuronal death and that alpha-synuclein aggregation.
So MP flips the mTORC1 switch too hard or keeps it jammed on.
That seems to be the idea, and the practical therapeutic implication of knowing this.
If you can block mTORC1.
Exactly. By treating the animal models with an inhibitor compound that specifically targets and blocks mTORC1 activity, the researchers were able to significantly reduce the neuroinflammation caused by MP. They could minimize the neuronal loss, lower the amount of alpha-synuclein aggregation, and ultimately significantly improve the motor symptoms in these Parkinson's model mice.
That is incredible. It provides a clear, actionable molecular target for future intervention strategies. It suggests that blocking this specific microbial-driven pathology, perhaps by inhibiting mTORC1, or maybe even by targeting S mutants or MP production itself, could be a key strategy for preventing or treating Parkinson's disease, especially maybe in its earlier stages.
It opens up completely new avenues, doesn't it? Moving beyond just managing symptoms to potentially targeting an upstream driver related to the microbiome.
Absolutely. So,
Yeah.
Okay, reflecting on everything we've discussed, what does this all mean for actual intervention? If dysbiosis, this imbalance in the gut, is so central to such a wide array of psychiatric and neurological conditions,
Yeah.
what are the validated or at least emerging strategies for intervention? What can we realistically do to modulate this complex ecosystem for better brain health?
Right. So what can we do question. The systematic reviews dedicated a significant amount of analysis to precisely this, therapeutic modulation. The main focus tends to be on probiotics, prebiotics, and then the more, shall we say, radical approach of fecal microbiota transplantation, or FMT.
Okay, let's start with the most accessible strategies first. Probiotics and prebiotics. Probiotics being introducing specific beneficial strains. Prebiotics being the foods that feed the good bacteria already there, like fiber.
Correct. And modulation with specific strains, particularly probiotics, appears highly promising for actually mitigating psychiatric symptoms, moving beyond just general gut health benefits like digestion. The studies consistently highlight targeted interventions using specific lactobacillus and bifidobacterium species. These two genera come up repeatedly.
Any specific examples stand out?
Well, one example was a robust study, randomized, double-blind, placebo-controlled, the gold standard, which found that administration of a specific strain, Lactobacillus rhamnosus HN001, to women during pregnancy and postpartum significantly reduced their self-reported depression and anxiety scores in that crucial postpartum period.
Postpartum depression and anxiety, that's a huge issue.
It is, and this study demonstrated a potential preventative or therapeutic effect in a known high-risk group just by using a specific probiotic strain. It suggests a strong potential for preventing or treating established mood disorders with these kinds of targeted interventions.
That's quite specific. Does the research suggest benefits beyond treating existing conditions? Maybe for resilience?
Yes, absolutely. The research even touches on psychological resilience. There was another interesting study on healthy young adults. They tested a multi-species probiotic formula, so a mix of different beneficial strains. They found that taking this probiotic significantly reduced their cognitive reactivity to sad mood.
Cognitive reactivity, what's that exactly?
It's basically the tendency to fall back into negative thought patterns or ruminate when you experience sadness or a low mood. It's a known risk factor for developing depression. So, reducing this cognitive reactivity suggests a potential prophylactic benefit, using probiotics not just to treat existing disease, but potentially to strengthen emotional regulation and make people more resilient against future stressors.
Fascinating. Building mental resilience from the gut up.
Oh,
Now, what about fecal microbiota transplantation, FMT? This is the most direct, maybe most radical form of gut modulation. Historically, it's been reserved for serious recurring infections like C. difficile. Is it showing measurable promise in the psychiatric context too?
The preclinical evidence for FMT in this context is both very strong and, frankly, quite provocative.
Right.
There have been studies involving FMT where gut microbes from human patients diagnosed with specific psychiatric conditions. They've used donors with depression, alcoholism, even anorexia nervosa have been transferred into those germ-free mice we discussed earlier.
Transferring the human patient's microbiome into mice.
Yes, and the recipient mice, the ones getting the transplant from psychiatrically ill donors, subsequently exhibited the behavioral phenotypes, the symptoms, essentially, of the human donor. They showed anxiety-like behaviors, depressive-like behaviors, impaired weight gain in the case of anorexia donors. It confirms the direct transmissibility of complex psychiatric traits via the microbiome in these animal models.
That is extremely powerful evidence for causality, isn't it? It strongly suggests the psychological state is tightly bound, maybe even encoded in some way, within the structure and the chemical output of the gut microbial community itself.
It really points in that direction. Now, clinically, in humans, FMT is showing promising, though perhaps less consistent, results for improving symptoms of depression and anxiety. This is often observed initially in patients who are undergoing FMT for other reasons, usually comorbid gastrointestinal issues like severe irritable bowel syndrome, IBS, or inflammatory bowel disease. Intriguingly, some studies report that these psychological benefits, improvements in mood or anxiety scores, sometimes occur independently of whether the patient sees significant improvement in their core gastrointestinal symptoms.
So the brain benefits even if the gut symptoms don't fully resolve?
In some cases, yes, which really underscores the potential for brain-specific effects resulting from the microbial community change induced by FMT.
That sounds like a potential breakthrough, especially for difficult cases. But when we discuss a major medical intervention like FMT, we absolutely have to consider the practical realities and the downsides.
Oh.
What are the known limitations or concerns regarding FMT as a psychiatric treatment right now?
The limitations are significant, and it's crucial we acknowledge them. FMT is not a magic bullet yet. While it offers this powerful, conceptually different alternative, especially perhaps for treatment-resistant patients who haven't responded to standard therapies, the therapeutic effects can be transient. They don't always last.
Transient. How long do the benefits typically last?
Some studies show the benefits, like improvements in quality of life or fatigue scores among patients with IBS who received FMT for their gut issues, often start to wane after about 6 months. The original microbiome might start to reassert itself, or the underlying conditions driving the dysbiosis haven't been addressed. Furthermore, the procedure itself is inherently labor-intensive. Finding and screening donors is complex, the preparation is involved, the delivery method, it can be costly. And critically, because it involves the transfer of living biological material from one person to another, its long-term safety profile, especially when used for chronic non-infectious conditions like psychiatric disorders, requires much more rigorous and ongoing investigation. Standardization is still a major challenge. It's not ready for prime-time widespread use in psychiatry just yet.
Right. Safety first, always, especially with transferring a whole ecosystem.
Exactly. Much more research is needed there.
Okay, we've covered a truly massive amount of ground today. We've journeyed from the basic anatomy of the gut-brain axis down to the molecular messengers, the SCFAs, the neurotransmitters and neurotrophins like BDNF. We've seen the clinical correlations across a wide spectrum of psychiatric and neurological diseases, and we've touched on the emerging interventions. Bringing it all together, what is the final essential synthesis? What's the big picture takeaway we absolutely must grasp from these comprehensive reviews?
I think the profound shift in our understanding of brain health is undeniable. That's the core message. We absolutely must now view psychiatric and neurological conditions not as purely brain-centric events, but as systemic processes that are heavily influenced by gut ecology and the resulting chronic, often low-grade inflammation. The most consistent and unifying finding, the thread that ties almost all of this together across nearly every major psychiatric disorder reviewed: depression, ASD, eating disorders, psychosis, even Parkinson's, is clear dysbiosis, an unhealthy gut microbial state.
And that dysbiosis has common features.
It does. It's typically characterized by that reduction in beneficial fermenting taxa, leading to a functional loss of protective SCFAs, coupled with an associated increase in pro-inflammatory or opportunistic bacteria. This imbalance creates a cycle that reinforces the core theory emerging from all this research: that brain status, its stability, its resilience, its very health, is intrinsically tied to the inflammatory processes and the metabolic signaling that are mediated by the gut-brain axis.
So the era of treating the brain as this isolated organ, completely disconnected from the rest of the body's metabolic and immune systems, that era is definitively over.
It's over. We are learning quite clearly that optimal brain function requires optimal gut ecology. The two are inseparable.
Which leads us to our final thought for you, our listener. Something provocative to consider based on today's deep dive. Given the extremely specific, identified mechanism we discussed, where a common oral bacterium, Streptococcus mutans, the one that causes cavities, produces a toxic metabolite, MP, that appears to directly contribute to neurodegeneration in Parkinson's disease via the gut. Given that, the future of preventive neurological health might depend less on finding complex pharmaceutical silver bullets and maybe more on the seemingly simple daily maintenance of our internal environments, our microbial ecosystems. If your oral bacteria and your gut bacteria are literally producing molecular signals that cross the blood-brain barrier to either protect or potentially harm your neurons, what does your current lifestyle actually mean for your long-term cognitive resilience? Think about your diet, your stress management practices, your use of antibiotics, and yes, maybe even your daily dental hygiene. What signals are you sending to your brain? The answers, it increasingly seems, start deep in your gut, and perhaps surprisingly often, right there in your mouth.